Okay, so in this video we're going to talk about the structure of nerves and associated ganglia. So a nerve is just a cord-like organ of the peripheral nervous system. Ultimately, this nerve is made of a bundle of myelinated and non-myelinated axons, which are encased by connective tissue. And there's really two major types of nerves. You have spinal and cranial nerves, and we'll actually talk about the certain subtypes of these in future videos. But what's nice is that these nerves are actually encased in connective tissue coverings, which are named a lot like how skeletal muscle is packaged. So we find that endoneurium is a loose connective tissue that surrounds and encloses individual axons and their myelin sheaths. And perineurium is this coarse connective tissue that actually bundles axons into fascicles. And then epineurium is a tough fibrous sheath that surrounds the entire nerve. So if you look at this cross-section of a nerve in an electron micrograph, we find that a blood vessel in the middle, and this blood vessel helps to nourish the axons from the inside. And then we have this outer covering of epineurium, which is a fibrous connective tissue sheath that surrounds the whole nerve. We have nerve fascicles, which are bundles of axons, which are encased in perineurium, and then individual axons here, which are surrounded by endoneurium. So if we look at an artist's rendition of this, we see epineurium that surrounds the entire nerve. We have nerve fascicles here that are surrounded by perineurium, which you can see here surrounding this fascicle, and then individual axons that are either myelinated or unmyelinated. Either way, they have an endoneurium, which is an areolar connective tissue that surrounds and nourishes this axon. So most of the nerves in the body are mixed. Now, when we say that these are mixed nerves, we say that they actually contain both afferent and efferent axons. So it's carrying both sensory and motor information. And there are some nerves in the body that are only sensory or afferent or only motor or efferent. However, these are more common in the cranial nerves. So pure sensory afferent or pure motor afferent nerves are pretty rare because most nerves are mixed. But these nerves contain different fibers. And these fibers or axons can carry different types of information. We either have somatic and visceral information, and that can either be afferent or efferent. Remember, somatic means coming from the body, and by that we mean skin, muscles, and fascia, and joints. And if it's visceral, we refer to the guts. So we're talking about like internal organs and glands, or smooth muscle and cardiac muscle. So for the somatic afferent, this is sensory information from muscle to the brain. If it's somatic efferent, this is motor information from brain to skeletal muscle. And then the same goes for visceral, right? So if it's visceral afferent, we're saying it's sensory from organs to the brain. Or if it's visceral efferent, we're saying that it's motor from the brain to cardiac muscle or smooth muscle and glands. And these ganglia, which are part of the peripheral nervous system, are essentially neuron cell bodies that are associated with nerves. Ganglia are associated with afferent nerve fibers, contain cell bodies of sensory neurons. And so an example of where we find this would be like the dorsal root ganglia of your spinal cord, contains somatic sensory cell bodies, we talked about back in chapter 12. And then ganglia associated with efferent nerve fibers are typically autonomic motor neurons. And we'll talk about the autonomic ganglia, which go to the visceral smooth muscle, cardiac muscle, and glands. We'll talk about that in chapter 14. So central nervous system axons form these fibers that unfortunately don't regenerate that well. And these central nervous system oligodendrocytes bear growth-inhibiting proteins that prevent your central nervous system fibers from regenerating. And this is important because we don't want our brain to expand within the skull. So it's important to inhibit too much growth of the brain tissue. Now, astrocytes at the site of nervous system injury form scar tissue, and treatment of this is basically going to prevent scar tissue formation. That way the brain can heal better. Now, in the peripheral nervous system, these axons can regenerate better than the central nervous system. And this is provided if the damage isn't severe enough. So this occurs, in several key steps here. We see that after the axons are damaged, those fragments in the myelin sheath degenerate, and this spreads down the axon towards the cell body and away from the cell body. Now, macrophages clean up the dead axon debris, and Schwann cells are stimulated to divide. And the axon filaments grow through this regeneration tube, which is made of the Schwann cells. And as the axon regenerates, a new myelin sheath forms around that new axon. So if you look at this in a step-by-step manner, we see that if the axon is damaged here, the part of the axon that's closest to the cell body kind of pinches off here. And every part of the axon that's distal from the cell body or away from it, that actually degenerates. And what ends up happening then is that macrophages come in here and they start to clean up these dead axon debris, and then Schwann cells divide to form a nice little tube. And so what happens next then is that then the axon can regrow through this tube of Schwann cells, provided there isn't too much debris in the way, and it's guided by growth factors that are released by these Schwann cells. And you can kind of think about this new axon as forming, or following rather, the chemical scent trail of the previous axon, until eventually the new axon is reformed and a new myelin sheath is regenerated. Now, if this occurs properly, we should see no loss of function or only slight loss of function. So you might only see slight loss of sensation or motor output. And the peripheral nervous system is better at regenerating than the central nervous system. However, one of the challenges that our body encounters is that if the axon's damaged and there's scar tissue that's formed here, the new axon can't grow past that scar tissue, and that could actually prevent the reconnection of axons from, its side of tissue. And this is sort of what explains why in some people with nerve injury, they just never recuperate function, because if that scar tissue blocks the way, those nerves can never reconnect.